Great. So it's a great pleasure to be here presenting this work on the impact of pharmaceuticals on the human gut microbiome. So among the intrinsic and extrinsic factors that can modulate gut microbiome function and composition, medication is actually a very important one. In fact, there are studies that have shown that the influence of a drug on the gut microbiome can actually exceed the impact of the disease for which that drug was prescribed. And this goes way beyond antibiotic drugs. And we know that thanks to a landmark study that was published in 2018, where the authors screened a large collection of human targeted drugs. So these were drugs targeted to human cells, eukaryotic cells, and processes, not to microbes. And their results have shown that among the hundreds of drugs that they screened, that 24% of those actually ended up impacting microbial growth, in this case, inhibiting the growth of gut microbes in vitro. So among the classes of drugs that the authors tested, one that stood out was nervous system drugs, in particular antipsychotics, which seem to be the class of drugs that have the strongest impact in terms of inhibiting the growth of microbes. So when we started this project, what we wanted to understand is how do these drugs impact microbes, not just only microbial growth and gut microbiome composition, but also how would they impact microbial activity and metabolism, and also why were they having such an impact on microbes, or was the mechanism behind it? So we started by investigating two nervous system drugs, entacapone, which is used in the treatment of Parkinson's disease, and loxapine succinate, which is used to treat schizophrenia. And we have incubated freshly collected human samples, fecal samples, with these drugs at two different concentrations. A low drug, or a low concentration, sorry, which was the same concentration that I mentioned in this study that I just told you about, and a high concentration of each drug, which is the concentration that is estimated that these drugs would reach in the large intestine. And after incubation period, we have looked at things like total microbial loads, microbiota composition, but we took this one step further and we also looked at microbial activity. And we have looked at microbiota activity by supplementing our incubations with heavy water. So heavy water, or deuterium from the heavy water, can work as an excellent marker or tracer of microbial metabolic activity. And this is because deuterium from the surroundings or from the heavy water is incorporated by metabolically active cells, and using techniques like stimulated Raman scattering, we can, with high throughputs, detect those deuterium levels into a cell's biomass. So if we amend the incubations with a pharmaceutical drug, stimulated Raman scattering enables us to look at changes or decreases in microbial activity, because those cells would likely have less deuterium incorporation. So the first thing we looked at from these incubations with these two pharmaceuticals was how were they impacting total microbial loads. And you can see from the results that I'm showing here that it's very obvious that one of the drugs or conditions, entacapone high, was dramatically impacting the total microbial loads and microbial abundance. However, we did not see a major effect of any of the other conditions we tested, or at least they did not seem to be significantly impacting abundance. However, when we took a closer look at these samples with SRS, with the Raman approach, we could see that cells were displaying lower levels of deuterium incorporation, which are showing here in the y-axis, and you can see that in here, and that those decreases were actually significant. So these human targeted drugs, also, they were not impacting microbial abundance, they were having an impact on microbiome activity. So we then, of course, sequenced these samples. We started with 16S RNA gene amplicon sequencing, and we integrated the relative microbiome profiles, or the relative abundance profiles that we got from 16S sequencing, with this data on total microbial loads, and that enabled us to generate quantitative microbiome profiles for our samples. So what you're looking at in here is that the absolute abundance of different taxa that we detected in our incubations, and this data is from triplicates per condition at two different time points. And so when we looked at the entire table, we could see that there was one drug or one condition in particular that was having a major impact, and that was entacapone. And that is very obvious from this heat map here. So entacapone impacts up to 30% of all taxa that we detected in these incubations, which is quite a remarkable impact or effect for one dose of a medication in such a short period of time. The other conditions we tested did not seem to have such a strong effect. The effect was more moderate, but as I just shown you before, these drugs seem to have a stronger impact on microbial activity than they actually have on microbial abundance. So we decided to take a closer look at some of the taxa displayed here. And so to do that, we complemented our sequencing with metagenomics. So we did Illumina shotgun and long read sequencing, and that enabled us to retrieve many high quality mags or genomes for the taxa that we detected in these incubations. And because those genome beans contain 16S and 23S RNA gene sequences, then we could design fluorescently labeled oligonucleotide probes that were specific for particular taxa that we were interested in. So we designed those probes, we targeted those microbes with fish, and we complemented this analysis with SRS to look at the levels of deuterium and microbial activity. And you can see here the results for the 6 taxa that we targeted. And overall, when we analyzed all the data that we gathered, we could observe that key microbiome members like Clostridium species, Bacteroides uniformis, or Ruminococcus bromai were being negatively impacted in terms of their activity in the presence of the drugs. But again, the impact that we detected in terms of their abundance was not that pronounced. So with this in mind, um, we concluded that many of these drugs, although they cannot impact microbiome activity or microbiome abundance, they really impact the activity of key microbiome members. Another thing that was enabled by this Raman approach was that we could actually image one of these drugs. So entacapone has an intrinsic photothermal signal, and SRS was able to capture that signal. And so what you are looking at in here is that the signal in green of the drug, entacapone inside microbiome cells, which are shown here in red. And because we have these fish probes that could target particular taxa in these complex fecal microbiome samples, we then went to with the fish approach to see whether some of those microbial taxa were incorporating or were showing higher levels of entacapone bioaccumulation. And that revealed that faecal array seems to be one of the taxa that show the highest accumulation, but all of the taxa we targeted actually showed some entacapone bioaccumulation to different levels. Now, interestingly, some of these microbes are inhibited in terms of their growth or abundance in the community, but others are not affected by entacapone despite they bioaccumulated or are even increasing their abundance. And this is quite puzzling, or it was quite puzzling for us at the time, right? So why do we have a drug that is bioaccumulated and in some cases leads to growth inhibition of that microbe or to decrease activity and in others to higher activity and even pronounced growth? So to try to better understand mechanistically why that was the case, we took a closer look at the structure of entacapone, which is shown here. So entacapone contains these nitrocatical group, and catechol groups are known to bind iron with high affinity. They are very common in siderophores, caticulate type siderophores, which are molecules produced by microbes that can complex iron. So we ran the typical assay to determine if a molecule is able to complex iron, and entacapone did pass the test, as you can see here by this change in color. And this was actually, or this had actually been shown by others previously using different approaches. So entacapone-induced, due to the presence of this catechol group, can complex iron. Now, iron is an essential micronutrient for most microbes, especially for gut microbes. So they need iron and we use iron as a cofactor for enzymes, and it's key for multiple processes. So what we postulated was that because this drug is complexing iron, is depriving iron microbes, from accessing iron. And so it's basically inhibiting growth because microbes cannot access this essential micronutrient. So we went back to our incubations and we actually, in parallel to supplementing them with entacapone, we have also supplemented our samples with entacapone and an excess of iron. More iron than what actually the drug could complex. We also supplemented these incubations with entacapone pre-complex with iron, and again, meaning that entacapone would no longer be able to complex any free iron that was in the incubation vials. And we could indeed see, as you can observe here from the blue lines, that we have a partial or a near complete rescue of the impact that the drug alone had on the microbiome when we had this excess of iron. And this was true not only for total microbial loads, but also in terms of the abundance of key taxa that were being impacted by entacapone and that are no longer impacted in such a magnitude when an excess of iron is added. So mechanistically, this seems to be a reason why entacapone is having such a pronounced effect on the microbiome is due to its ability to deprive microbes from iron. So at this stage, there was only one piece of the puzzle that we were missing, and that was why is it that some microbes seem to still survive and even thrive in the presence of entacapone? If iron is an essential micronutrient, how are they doing this? So one of those microbial taxa that was particularly enhanced in the presence of entacapone was Escherichia shigella. We have only, this was actually E. coli, and we have only one E. coli strain in our incubations for which we had retrieved a near complete metagenome assembled genome. So we did into this metagenome assembled genome, what we could see is that this E. coli strain contains or harbors a complete operon for entrovectin production. And entrovectin is a siderophore that is able to complex iron with higher affinity than entacapone. So our conclusion was that was exactly what was enabling this microbe to thrive in the presence of the drug, was that it was able to produce other molecules that would scavenge the iron that entacapone was taking away. And we confirmed this later on with a series of tests using genetic mutants for entrovectin production in E. coli. So with this, I'm already reaching the end of this talk. And so our main take-home points are that these two drugs that we tested have different impacts on microbiome composition, but both impact microbiome metabolism. And entacapone dramatically shapes the microbiome and inhibits the abundance and metabolic activity of many gut microbes in these samples. And this is because it complexes iron, depriving microbes from accessing these essential micronutrients. And selecting for microbes that are iron scavengers, that are able to produce siderophores or other proteins or other molecules that have a high affinity for iron. Of course, all of this was done in vitro or ex vivo, and we are now trying to follow up on this to see whether the effect of this medication in patients, in Parkinson's disease patients, actually is confirmed. And if so, this is potentially worrying because we see that many of these iron scavengers that are basically selected by the drug have virulence genes and also a higher number of antimicrobial resistance genes. So potentially entacapone is selecting for pathobionts. And if we observe that this selection also occurs in patient cohorts, then we can propose an iron supplementation together with the entacapone therapy so that we can avoid any side effects or collateral effects that the drug is having on the microbiome. And so before I finish, I would just like to thank the team that was behind this work. This was really a great team effort. The work was published recently or came out last November. So please have a look if you're interested on knowing more of the details. And thank you all for listening. And I'll be happy to answer any questions in the Q&A session. Thank you.